Good morning, everybody. Welcome to the MPG primer today. It is my pleasure to introduce Pamela. Pamela is a genomic specialist for ClinGen at the Broad Institute and an expert in variant and gene curation. She co-chairs the ClinVar curation working group advancing accurate variant classification. Her background includes germline and somatic curation for an Australian childhood cancer program and a biotech startup, where she also developed genomic training workshops. Pamela serves on the board of directors for the Federation of American Societies for Experimental Biology, the Education Committee of the Cancer Genomics Consortium, and the Professional Relations Committee of AMP. She earned her PhD studying microRNAs in head and neck cancer. And today she's going to give us a primer on clinical genomic resources. So thank you very much, Pam, for speaking today.
Thank you. I want to start off first by saying it's my pleasure to be here today. I could talk about ClinGen, ClinVar, genomic resources all day. I won't do that though. I will stick to the 45 minutes. But before I get into the deep dive on genomic resources, I wanted to start off first with a story. So this is a story about a disease that's been a defining challenge in medicine for over the past century, which is sickle cell. And this is a life-threatening genetic disorder that's associated with anemia, infection, and episodes of pain. If you look at the figure on the right, it shows how red blood cells are in the sickled cell shape, hence the name, that can cause them to get stuck in blood vessels, blocking blood flow and leading to really serious downstream health consequences. Now, this is caused by a single base pair change in the hemobeta-globin gene, and an individual with sickle cell disease has both copies of that gene mutated, and this is the most common variation of sickle cell disease. So it is known that it affects millions of people throughout the world and is particularly common among those whose ancestors came from parts of the world where malaria was or is still common. And so sickle cell is a really classic example of a monogenic disorder where you have a change in a single gene that leads to really profound clinical consequences. Now, in 2023, the narrative did change, and some of you might notice. So what I've got here is a press release from the FDA where they had approved 2 milestone treatments. So this was Casgevy and Lyfgenia, which were the first cell-based gene therapies for the treatment of sickle cell disease. Also really cool is that Casgevy was actually the first ever FDA-approved therapy that was developed with CRISPR technology. And it's my understanding it's actually the work of one of the Brodies that also contributed to this. So that's just a really great example of seeing that bench to bedside happen in action. Now, I do have a bit of a personal connection with this disease. I actually recently found out through carrier genetic testing that I have sickle cell trait, which means that I carry one abnormal copy of the beta-globin gene. And the genetics behind that is that I have what we call a missense variant. So in the gene, on the DNA level at a specific position, instead of an adenine, I have a thymine, so going from A to T, and on the protein level, that's going from glutamic acid to valine. What does that mean in terms of symptoms? Honestly, I have never really felt any symptoms throughout my life. I wish I had known about it a few years ago when I was in Bali, worried about mosquitoes and malaria, and not knowing that all along I kind of had this superpower, essentially. There are some serious implications if you are planning to have a family. So if my partner is also a carrier, and I've just got a little diagram down there, then our child has a 1 in 4 chance of developing sickle cell disease. I'm going to be honest, we weren't too worried just because my husband does look like this, but it's not 0, so we still got him tested. And what I want to now show is our first genomic resource, which is Nomad. So this is a huge resource of the general population with exome sequencing and genome sequencing data. And you can essentially look at a variant and see, is this a rare variant or is it common in the general population? And you can even look at it further by ancestral groups. So I've highlighted two. I'm in the African-American bucket, where the frequency for the variant that I have is about 5%. Actually turns out, because I have generations of Nigerian heritage, it's more like 25% for me. For my partner, who's of the European non-Finnish background, you can see it's quite low. It's like 0.004%. So it's always worth to check and do your genetic testing if you can. But it really starts to be, this is my segue into the rest of the talk, showing genomic resources, why they're important, how we use them. So there's heaps out there, lots of resources. I'm just going to speak about some of them in a broad level and just show some examples as well. So grouping them into three different buckets, we've got like the gene level associations, where we're trying to figure out, is this gene actually linked to this disease? And you can see some examples along there. And then there's the variant level side, where you want to know, are there interpretations that have been made about this variant? Is there any kind of functional data that can be used, or allele frequencies in databases like Nomad? These can be publicly available, so anyone can access that information, or it can be commercial, where you got to pay a little bit of money to access that database, or in some cases, a lot of money. And then there's the case level side, where you have de-identified patient information, and you're looking into that data, identifying variants and the phenotypes, the observable traits, and trying to make associations there. So it is a ClinGen, ClinVar, other genomic resources talk, which means there's going to be a heavy talk on ClinGen. So this is short for the clinical genome resource. And it's really, the purpose behind it was to, and is to, create an authoritative central resource that defines the clinical relevance of genes and variants for use in precision medicine and research. This started back in September 2013. And it is primarily funded by the NIH, which in 2025 in the States, that's a fun time, as I'm sure you're all aware. It's covered by 3 core grants throughout the US, with PIs at multiple sites, including at the Broad, 1 somatic cancer grant, 1 pharmacogenomics, and just over 20 disease-focused grants as well. And all of this is fueled into this mission on the left, which is an infographic depicting what we do. So we have our stakeholders at the top, where we're able to aggregate de-identified genomic and health data, and use that to answer ClinGen's 4 critical questions. 1, gene disease validity. Does variation in this gene lead to disease? 2, variant pathogenicity. Is this variant disease causing? 3, dosage sensitivity. Does copy gain or loss of this gene or genomic region lead to disease? And 4, clinical actionability. If this gene was significantly altered, could it confer a high risk of serious disease that could have been mitigated if that disease risk was known? And so we do this in the form of our expert panels, is what we call them, which consists of scientists and clinicians around the world who are experts in the field, and we come together on a frequent basis, focusing on a series of diseases and answering these questions using established frameworks that have been created. And then this information is disseminated to the public, and it fuels back into these questions, and we have this feedback system. And the end goal here is really just to improve patient care through genomic medicine. Additionally, ClinGen also has the honor of being a global core biodata resource, meaning that it is considered of such fundamental importance to the biological and life sciences community for the preservation of biological data. So it's a cool honor to have. So this paper came out last year from the ClinGen Consortium, and really just shows the progress of the past decade in essence. And the figure on the right really shows the explosive growth of ClinGen's expert panels over time. You can see in pink the gene curation expert panels, and in blue are the gene curations. And then in orange are the variant curation expert panels, and in green are the variant curations. And we're now at what we call phase 3, which is all about scaling and sustainability. So we have so much data. How do we continue to maintain this in a feasible manner? And also, how do we scale our efforts? There's still so much to do, and how do we do that in a resource-sensitive and mindful way? Here I've got a slide showing ClinGen leadership, who are responsible for oversight and the direction of ClinGen as a whole. And they're all around the US. And then additionally, we also have 100 plus staff members such as myself, who are involved in additional leadership roles, coordination. ClinGen is a beast. It's a fantastic beast, but there's lots of moving parts, so coordination is necessary. Biocuration, software development, and many other roles. We also have over 50 working groups that manage standards, operations, and software, and also over 140 expert panels. Wanted to highlight the ClinGen team at the Broad site, which is where we're at today. So this is co-led by the PIs. You can see here Professors Heidi Ream and Marina Di Stefano at the top. And then this is a diligent, hardworking team that really makes sure that we keep ClinGen running from our side. Okay, so let's start to talk about ClinGen's expert panels. So in order to do that, we have what we call our clinical domain working groups. So there's 15 of them. You can see them on the left there, focus on specific disease areas, essentially. And then that leads to 58 gene curation expert panels and 76 variant curation expert panels. And another thing that I really want to highlight is that, yes, we have about 100 or so staff, but the bulk of ClinGen is actually our 2,800 plus volunteers all around the world from 74 countries that actively contribute to our expert panels and our working groups. So ClinGen wouldn't exist without them. So we're always very thankful for that. So today I'm going to be focused on gene and variant curation. I'm not going to talk about dosage, clinical or somatic. But before we can define a variant's role in disease, we first must assess the gene level evidence. And so I want to show another resource, which is OMIM. This is an absolutely comprehensive resource for finding claims that have been made between a given gene and disease. Looking at recent stats from OMIM, they have over 7,000 phenotypes or observable traits for which there is some sort of molecular basis known, which equates to about 5,000 or so genes that are associated with a phenotype causing mutation. So that's a lot of data, especially for very other comparative resources that do the same thing. In order for a gene to enter this database, it does need a minimum level of evidence. So it's not just anything and everything. It's really genes that it seems like there could be something there. And OMIM includes variants for a given gene and disease that are kind of like exemplar variants for that. So it's not a complete catalog either of every variant that has been reported in association with that disease. Now, there's lots of ways to use OMIM. I'm just going to show a really simple way where you can put in a gene. And I've put in here MSX1, and you can have a look and see, okay, what diseases have been asserted for this gene. And then you can click into them and look into it further and see, okay, what are the phenotypes associated with this disease that have been reported? If you've got patient data you're looking at, does it match your patient? If you're trying to figure out more about the disease and figure out if it's part of a spectrum, you can do comparisons there. There's lots of different things, and it's a really great starting point for that. Okay, so now I'm going to start to talk about gene disease validity, which I'm using interchangeably with gene curation from the ClinGen side of things. So on the top left there is the publication that came out a few years ago describing the evidence-based semi-quantitative framework that ClinGen uses for gene curation. And essentially, this is subdivided into genetic evidence and experimental evidence. So when we're thinking about genetic evidence, we're thinking about things like case reports, case series, case control data, family pedigrees, and we have a point system that we use as a scoring metric. And then similarly with experimental evidence, we're looking at things like biochemical evidence, expression data, animal studies, all of that to support the gene-disease relationship as well. And so the points are added up, and then based on the amount of points you get, the more points you have, the more evidence supporting that gene-disease relationship. So at the top, we have definitive, if I can get my mouse to go there, as the most amount of evidence, and then at the bottom, we have refuted as the least amount, basically saying that at this point, there is no substantial evidence at all supporting that gene-disease relationship. Okay, so there are implications downstream on what the final class is for a given gene-disease relationship. On the right, technical standards that came out a few years ago from the American College of Medical Genetics and Genomics, ACMG, basically giving guidance around how to diagnose a sequencing panel and report variants from that. Now, in essence, if you are in the kind of discovery stage, research settings, you're going to identify genes and diseases that meet that entire spectrum of classification. However, as soon as patients are involved, you need to have a pretty good amount of confidence that that gene is actually associated with that disease. And so when you want to design a diagnostic panel, the recommendation is to have moderate or above as the evidence strength for that gene-disease relationship. And then the next tier above that is predictive testing, where you have an asymptomatic individual who wants to understand their risk of developing disease. And so for that, we use the highest tier of evidence for gene disease validity classification. So this is just showing the data from ClinGen's 58 gene curation expert panels. That's just a subset of them there, color-coded by the final classification that they received. When I last checked, it was 3,331 total gene disease curations. 3 is my lucky number, so I was kind of hoping it'll be 3333 by the time this talk came around, but no such luck, that's okay. And then this is just showing at the top the distribution of the classifications. And you can see the vast majority are definitive. However, there is a subset of the insufficient evidence, about 23%, which don't have enough claim. And that's kind of concerning because a lot of the times when we're looking at these genes in our expert panels, it's because they're on diagnostic panels. So we shouldn't be seeing them, you know? They should be having more evidence. So it's a little bit concerning, and it just highlights why it's important to do gene curation first. All right, this is our golden example of like why you need to do gene curation first. So this was a paper that came out a few years ago from the Brugada syndrome expert panel. They were looking at 21 genes that are commonly found on diagnostic panels for Brugada syndrome. So they applied the gene curation framework, and lo and behold, to their shock and horror, only one gene, just one gene, had definitive evidence for Brugada syndrome. The remaining 20 genes were actually disputed, meaning that the evidence was conflicting at best. And yet these were appearing on diagnostic panels, right? That's a worry. And so it just really highlights why we really encourage gene curation to be done before designing a diagnostic panel and using it on patients. So these are just a few screenshots showing the ClinGen knowledge base. It's honestly quite huge. There's no way I could show everything in time, but there's lots of different uses. If you haven't used or been on this website before, it's a treasure trove of information. I recommend you use it. I'm not just saying that as someone who's paid to work for ClinGen. I said that for years prior, even receiving a single paycheck from the place. But it is genuinely a fantastic resource. You can put in your gene of interest and you can see if there's any kind of curations in the gene space, the variant space, the C and V space, somatic, clinical actionability, all of that, and see what evidence that has been expertly curated towards it. So everything we do, we're all about being thorough and transparent. And so when you click into any gene, disease, or variant, you'll find out the evidence that we use to score it, how we scored it, and why. So just here is just an example of the MYH7 gene. And you can see from the gene disease validity perspective that it has been curated against 4 different diseases. The final classifications are different, especially for that last one there. And you can click in further and find out why. If any kind of CNV review has been done, that will appear under dosage sensitivity. And then similarly, actionability will have a report if that's available. I'll talk about our variant curation expert panels later, but if one exists and they've created specifications and curated variants, you'll find that evidence there as well. So I want to talk about the curation ecosystem now. So I've talked about ClinGen quite a bit and the consortium and what we do, but we also work quite closely with 2 other resources. The first one I'm going to go into is GenCC, or the Gene Curation Coalition, which is a database for sharing gene-level knowledge, and this is supported by ClinGen. So GenCC is actually co-led by 2 of Broad's PIs that I introduced earlier, so Marina Di Stefano and Heidi Ream. And so their vision before GenCC became a reality was based on the fact that for a while, you had lots of groups out there who were also doing gene curations, all in isolation and not sharing their data. And as we know at science, collaboration is key, right, for scientific progress and discoveries. And so they envisioned essentially what could be a one-stop shop for gene curations and convinced quite a good number of submitters, which you can see up there, to come together and share it in this database. So this is a combination of public and private resources. And part of creating GenCC and getting them on board was also developing consistent terminology for gene disease validity assessments. And if you think about it, right, if we're calling something with the most amount of evidence definitive, another group needs to have the same name, or we need to come to an agreement as to what that name would be, right? We need to be speaking the same language. We don't want to be comparing apples to oranges. And so this has led to the public sharing of not just published curations, but also unpublished curations from these groups. And they now meet frequently to collaborate and resolve discrepancies on gene curation. So in terms of overall stats from GenCC, they have over 24,000 submissions in their classifications on over 5,000 genes, so very similar to OMIM, and 16 submitters that actively contribute their gene curations. And on the bottom, you can again just see the distribution of those classifications, where similarly to ClinGen, about 17% of those have insufficient evidence supporting the claim. So one thing that you can do with GenCC, there's lots of things actually, but you can put in like your gene of interest or disease, or you can even look by submitter, and basically you can see what information is out there. In this example here, we have GJB3, and you can see that it's associated with 8 diseases from 7 different groups. And so you can just like have a look and see, okay, what are the submitters, diseases, and what classifications did they come through, and what's concordant or discordant between them. So I want to move on now to variant pathogenicity.
Can I ask a question before you move to the next part? Online, someone is thanking you for bringing up OMIM, and they just wondered why there's no OMIM numbers, or if it's possible to add OMIM numbers to ClinGen.
As in our ClinGen curations also have OMIM numbers embedded within them.
That's what it sounds like.
So you can actually see in one of the sections where if you click down, it'll say what disease entries were used, and so OMIM links would be in there if used. All right, so where was I? Variant curation. I need to go back one slide. There we go. Okay, so when we're thinking about variant classification, there's a few considerations, right? So you want to have a set of common standards for variant classification. If you think about it, you want a group in Japan or a group in Australia or in Germany to see the same variant from a patient and review the evidence and come to the same conclusion about the variant's pathogenicity, right? In order for that to happen, they need to be using the same framework. Additionally, you want them to have access to the same pool of evidence. Similarly, if you have one group that has access to internal patient data, another group that has access to some functional assays that Jay just have, another group who has access to neither, for sure, they're going to come to different conclusions on the variant's pathogenicity. And so what we really want to encourage is a breakdown of those silos and the sharing of evidence so that we can try to come to a harmonized classification on a given variant. And so part of that is having a shared evidence base, which I will talk about in a bit. We also want the public sharing of variant classifications, right? If there different groups have different views and there's discordance, we want to know why so we can see what the discrepancies are. And then finally, we want to have expert consensus on these classifications. One, to help resolve discrepancies, but also for those groups who don't have the bandwidth or the resources to go and do those deep dives into the evidence to support them, and which is what our ClinGen variant curation expert panels do. So I'll get into all of that, but you can see on the right, just a subset. There are so many resources out there and they just keep growing day by day. But these are some very commonly used ones for variant classification purposes. Okay, so common framework. So we have the ACMG AMP standards, and the paper down there is just showing the paper that came out in 2015, so about a decade ago, which really is a cornerstone of variant curation. And describes this common framework. It acknowledges the complexity of curation, right? Classifying variants isn't black and white. I mean, sometimes it is, but a lot of the time it's not, right? It can really fall on a gradient. And so they developed this 5-tier classification system where you had pathogenic, likely pathogenic. So these are variants that are considered reportable and you can return them back to the patient. There's a variant of uncertain significance, or the VOSs, and anyone in this space would know they are the bane, absolute bane of our existence, and how labs can treat reporting them can be quite different depending on circumstances of the patient that they're dealing with. But there is guidance coming out to help with this, fingers crossed. And then we have likely benign and benign, which is considered not reportable because the variants are not disease-causing. So this is the table of evidence that we use for variant classification. It summarizes things like, is a variant common or not in the general population, like I showed with NOMAD? What do the computational predictors think of this variant? Are there functional assays out there? What about the family data? What is that showing? So using all those pieces of evidence to then combine them and come to a final classification, which is shown on the right. Now, those combining rules have over time proven a little bit tricky to remember. So we have started to shift towards a point system, which is quite intuitive. And we're expecting next year, we've been expecting this for a while, but we've been told it's for sure next year. We are having a long overdue update to the 2015 standards, and everyone's waiting with bated breath. And this should really just resolve some of the issues we've had with the current standards, such as subjectivity of interpreting some of the evidence. So with ClinGen's variant curation expert panels, or VSEPs, their goal is essentially to take those 2015 standards, which are generic in nature, and that's intentional, so that you can plug in a given disease or gene, but they take it and they're focusing on a disease area and a set of genes, and they capture that information and account for the biological nuances, right, that are associated with diseases. So that's what they do. It can take a long time, right? It can take a couple of years at least to put that together. And then those specifications, once approved, are made available by ClinGen for public use. So genetic testing labs, research labs can just go on, get that information and use what they need. Like, okay, what is the threshold of population frequency to consider this rare or frequent? Like, they don't need to do the research. We've done that for them. What we also do is that VSEPs then publish curations on a database called ClinVar to classify difficult variants and resolve discrepancies. So it's on ClinVar, but it's also can be found within ClinGen. One of our portals also takes us to that. The work of ClinGen VSEPs is a FDA recognized process because of how thorough our curations are and how transparent we have to be in terms of what evidence we used and why. So at this stage, we have 38 fully approved VSEPs associated with 89 gene disease specifications, with 82 VSEPs in total in various stages of development. So this equates to almost 12,000 variant curations that have been published. And this is just showing down here the different classifications that they've met. So going back to the curation ecosystem, I now want to talk about ClinVar. So this is a database for sharing variant level knowledge and is supported by the National Center for Biotechnology Information. So ClinVar has been around for as long as ClinGen, and at this point has over 6 million submissions, and that number keeps drastically changing. I feel like every time I talk about ClinVar, it's 3 million, 4 million, 5, now it's 6 million. And on 4 million variants and over 3,000 submitters from 92 countries. So the amount of data in here is unbelievable. By far, the vast majority of submitters are clinical labs, as you can see. And then a smaller subset are researchers, ClinGen expert panels, patient registries, and clinics. So over time, ClinVar has really proven to be a critical resource for the global clinical and research genomic communities. Very quickly, just for anyone who's not familiar with ClinVar and what that looks like. So this is just showing a variant record for a missense variant in the MYBPC3 gene. And I'm going to try to click her and see how I go here. Each row is a different submitter. Right? So on the left, you have information like what classification they gave the variant, if they used a framework, what we call the assertion criteria, they provide that, what condition it was curated, classified for, and who the submitter was, and when that information was put in ClinVar and if it's been updated. If they have evidence, that is shown. So if you see the second submitter, we have it under comment. We love to see evidence, by the way. It's important to know why a group came to a classification and what evidence they used. And then all this information from the submitters is then aggregated at the top to a final classification. And ClinVar uses a review status to indicate their level of confidence on that variant record. So if there's, and the review status is just at the top there. Okay, let's see. There we go. Okay, so if you get 0 stars, it means that no assertion criteria was provided, so you don't know what framework they used to classify the variant. They get 1 star if only a single submitter provided a submission, or if multiple submitters did, but they disagree, which is the case here, where you can see it's conflicting classification. So that gets a 1 star. 2 stars if multiple submitters agree, 3 stars if it's a ClinGen expert panel because of our FDA status, and then 4 stars if it's a practice guideline like CFTR in cystic fibrosis. Now, if we start to think of the landscape of variant records in ClinVar, the vast majority of them only have a single submission. It's about 76%. 9% of these are actually associated with conflicting classifications. And of that, 1%, which equates to 44,000 at the time, because this is based on the June data set from ClinVar, 1% of these are considered clinically significant conflicts, right? These are the ones that kind of worry you a little bit. They're the ones where in a variant record, you have a submitter or submitters that think the variance is reportable, path likely path, and then submitters who think actually the evidence isn't enough. It shouldn't be reported. It's a VUS, likely benign, benign. So you can understand that that would be quite confusing to someone who's gone onto ClinVar, looked at the variant and thinking, so what is it? Which one is it? And when you look at it, subdivide further on the top right, the majority of those clinically significant are actually at the border of being likely pathogenic versus a VUS. So the ones where like there's just enough evidence, but also for some not just enough. Like it could go either way. And those are the ones that really can be quite challenging to know which bucket they should be in. So ClinVar's goal was never to actually try to pass any judgment on the accuracy of the submissions that they receive. They just house them, and neither were they planning to review them over time. But it's become clear that some submissions are just inaccurate or just really old and are missing newer evidence that has come out. And so there's been calls over time to essentially clean up ClinVar. So that's where ClinGen steps in. It will step in a moment. Let me do it from the computer. Okay, there we go. So this is done in the form of what we call the ClinGen curation of ClinVar or CVC working group. So this is Heidi's brainchild, this project. And it's a group that her and I co-chair together, where we review old and outlier submissions in ClinVar to improve the accuracy of the content in there. And this is just revealing the team that spent a lot of time on this project. It's complex, so it does require quite a bit of scientist power behind the scenes. And it also includes Melissa Landrum, who's the PI of ClinVar. So it's something we really do hand in hand with them. So a little bit more about this project. So we're focusing on really obvious issues here. We're not trying to do any kind of variant classification. That would be a mammoth task for such a small team, and also somewhat redundant when we have our variant curation expert panels. What we're looking at are things that are what we consider clearly inaccurate, right? So a submission, in order to meet that category, typically needs to meet two qualities, which is that it's either quite old, it's an outlier, so it's causing conflict, or it's lacking supporting evidence. And so we review those submissions, we apply a flag, those flag submissions are passed on to ClinVar, ClinVar passes it on to the submitter, and basically the submitter has 60 days to contact us or update their submission with more evidence or change their classification before the flag goes live on ClinVar. And so what happens is that once the flag is live, the submission is pushed down to the bottom of the variant record. It doesn't disappear. That information in there can still be useful. It's just pushed to the bottom, and the classification is removed from the overall classification. So there's an example down there where you can see the flag submission. And so that was a VUS that was causing conflict, but you can now see after being flagged, the classification has now gone to just path, likely path, essentially, and gotten a 2-star. So this project has been going on for a few years, but it officially went live just over 2 years ago in ClinVar. And so in that time, almost 18,000 variants have been reviewed, leading to just over 4,000 variants containing a flag submission in ClinVar. So that's the amount of variants that have a flag submission. But in terms of the amount of actual flag submissions, it's 5,222. And what we've done, because we want to know what the community thinks of this, right? This is a community database, so we want to make sure they're receptive and that this is something they feel is working and useful. And so we've kept an inquiry log to keep track of issues. If there are any, ClinVar will reach out to us and connect us with the submitter. We then investigate on our end and respond. And so out of the 5,222 submissions, only 26 of them actually were challenged by 21 labs, and challenge is too combative. They really were just like, hey, why was my submission flagged? So that's about 0.5%. And a lot of those were actually in the early days. And so out of those, we retracted 7 flags on our end, and then the remainder, the submitter updated their submission with more evidence. But again, that was more at the start when we were dealing with some of the teething issues, as you can imagine, with a complex project. So it still appears to be really well received by the community. And we're now at the stage where we're trying to think, how do we scale our efforts with this project? If you do find an error in ClinVar, please send us the variant. We're always happy to review them. We have a request review form on the webpage that you can click in and put in your the information and details about the variant. So in the last little bit of time, I'm going to go through two very quick case studies in the application of the CVC project. One of them, you need to understand that there is a relationship between gene curation and variant curation. Gene curation needs to be done first. And the reason for this is that if you do curation, gene curation, and a gene gets limited or below, variants in that gene should not get, for that condition, should not get anything above a VOS. If it scores moderate, the cap there is likely pathogenic. And if it gets strong definitive, then the full spectrum of pathogenicity can be used for the variant. So this is an example of a gene curation on the FANC-M gene that was done by our cancer expert panels. There's 3 diseases, 3 different classifications. I'm using the Fanconi anemia as an example where this received a refuted classification, meaning variants in this gene for that condition shouldn't get beyond a VOS. When we went and looked in ClinVar, that is the case for the vast majority of submissions, but there is a smaller number that have classified their variants as pathogenic and likely pathogenic. And so this is obviously very confusing for outside people who are using ClinVar and looking at Fanconi anemia. And so we have a side project going with the CVC work where we actually review these low evidence gene disease relationships and see if there are likely path submissions in ClinVar that need to be flagged when appropriate. The second case study is from the Rare Genomes Project, which is at the Broad, and it's a patient-driven research study led by genomic experts and clinicians using genomic sequencing data to find answers for families with rare and undiagnosed conditions. And so in this example, we had 2 siblings here, a male and female in their 30s, with a list of phenotypes that you can see there. And we used Seeker to analyze their genomic sequencing data. Quick plug for Seeker for anyone who's not familiar with it. It's a web-based resource. It's publicly accessible, and it's fantastic if you have genome exome sequencing data for a given patient. You can put it in there, and there's some preset filters that you can use to try and identify, what variants could be causing the disease being seen in the phenotypes in the given patient. So we used that, and from that we identified variants in the KIF BP gene. When we look at GenCC, there are multiple submissions, including ClinGen, saying there's a definitive gene-disease relationship for autosomal recessive Goldberg-Sprinson syndrome, which when we checked out that disease, the phenotypes were a fantastic match for these siblings. And so we'd found what we call a genotype-phenotype match here. I won't go into the variants too much in the interest of time, except to say that so there were 2 variants identified. The first one, a nonsense variant, had all the hallmarks of clearly being pathogenic. And in ClinVar, it was supported with multiple submitters saying, yep, that's pathogenic. The second variant was a little bit trickier. The team was able to put enough evidence to get it at likely pathogenic, But in ClinVar, this was one that was causing a clinically significant conflict, with some calling it path likely path and one calling it a VUS. So this is a great case study for the CVC project. When you look at the path likely path versus VUS, we wanted to know why and what was going on there. So we looked at all the submissions, and the VUS is at the top, one of the likely path at the bottom. And basically, we ended up flagging the VUS essentially for 3 reasons. 1, it was an outlier. It was a lot older by several years than the other submissions. 2, it was causing the conflict. And then 3, it had insufficient supporting information. It didn't have as much evidence as some of the newer submissions did. So that met all the criteria for flagging the submission and removing it from the overall variant record. So I hope I've convinced you in this time on the power of clinical genomic resources. As I said, there's so much out there. Just chose a subset focusing mainly on ClinGen and ClinVar, but there is a lot out there. And they're absolutely essential for diagnosis and discovery. There's gene level, variant level, case level. There's also therapy level, which I didn't get into as well. I always have to do the final plug at the end. If you are interested in volunteering for ClinGen, there's the QR codes. We're always interested in having more biocurators and experts, but you can also find those forms on our website. And then I just want to finish off on this acknowledgement slide, including the Translational Genomics group, which is where the Broad ClinGen team is situated. Thank you. All this work is based on a consortium of people, so it is a lot.
Great. If you have a question, come up to the mic, and I'm going to quickly read one online first. For individual clinical labs, how do they get information for individual diagnostic test panels? Does each lab create their own panel, or is there some sort of central database or guidelines, for example, which genes or BRCA mutations would be included on a cancer panel?
So one of the most commonly used ones at the moment, and you can find that through GenCC actually, is PanelApp. So they have like a traffic light system for a given panel. So they have lots of panels in there saying, high level evidence is green, amber, yellow. I forget the 3 colors of the traffic system essentially. And so that's a great way to like get a preset panel. And then obviously you can also cross-check with GenCC to see if there's agreements about the final classification for those genes and the given disease as well.
Thanks for such a beautiful, clear talk. I have 2 questions. One is the kind of obvious one about how is AI going to transform all your processes? And the other one was, I've noticed in the screening world, people are like, well, actually, the model that we have of like, we've got genes linked to a disease, we've got variants linked to a disease is like not sufficiently rich, because you might think, people know, oh, but those variants in that part of the gene, they tend to be much less penetrant than these ones that are over here. Mm-hmm. So that I think there's like a felt need that we don't have a rich enough scheme to capture what we might want to pass on to patients, particularly in the screening absence of phenotype world. And I'm wondering if there are any thoughts about like making that underlying model richer.
So penetrance is a challenging thing to deal with in our space. And definitely for genes that really suffer from that. I can understand from a screening perspective if something has such low penetrance.
It's more a combination within the same gene. They know, the clinicians know that some are low and some are high.
Yes, agreed. And so, with the ones that have the high penetrance, right, there's less concerns there. It's the ones that are lower penetrant. And in terms of a framework, I should say that there is, so ClinGen does have a working group focused on like dealing with risk alleles and penetrance, and they've started to work on, and they've got a publication and a white paper that's come out, really providing guidance on how to deal with that in terms of classifying, but also reporting. So I think there's more to come in that space, but like agree that it's a common challenge, especially in the cancer space as well. And then, sorry, what was your first question?
What's AI going to do for you?
AI, it's the buzzword, AI. We are working on that, as I imagine every group is. And I think the hope is that a lot of what we do is so manually intensive And there's so many areas in there that really have the opportunity for automation, right? And using artificial intelligence to really do the work that we don't need to do. Like still have a human like overall review, but there's just so many things that are quite tedious. So we're looking into it. There's definitely lots of places along there that could be done, for sure.
All right. Thank you, Pamela, for a great primer.
Thank you.